Hey, so my name is Luke Perry. It is 11/17/2020, and I'm going to be doing my presentation on sex determination today. So let's start off by saying that there's three characteristics that differentiate males and females. That's your gonads, whether you're creating sperm through testes, or whether you're creating eggs through ovaries. That's the internal genitalia, which we'll get into. It's the tubular organs that develop in the fetus that are not the gonads or the external genitalia. The external genitalia consists of essentially the vagina and the penis. And we'll get into that more fully here in a second. So here's your external genitalia of males and females. Let's start with the males. Over here you have the scrotum. Right here you have the shaft of the penis and the glans penis. And so the glans penis and the clitoris from the vagina actually develop from the same structure here in the fetus. And so over here for our external structure for females, we have the vaginal opening. So the lower part of the vagina is there. The clitoris. The labium majus and labium minus. So we have that thicker skin around here, that is your labius majus, and then the inner skin, the inner flap of skin, is your labium minus. So now for internal structure. So internal structure, we have two ducts that we start out with in the developing embryo, and that's your Mullerian duct and your Wolffian duct. So for females, your Mullerian duct is a duct that develops into your uterus and the upper part of your vagina and the fallopian tubes. So that's really important. Whereas the Wolffian duct in the male over here, it develops into your seminal vesicles, your vas deferens, and your epididymis. And the prostate is there as well. However, that actually develops more fully with DHT, or dihydrotestosterone, and it controls the prostate development. And so right here we can also see our gonads. So the gonads right here are the ovary that produces follicles that eventually turn into that have oocytes in them. And then you have your testes, your epididymis. The testes actually have things called semiphenimers tubules that actually produce sperm with Sertoli cells and Leydig cells. We'll get into that more fully later. And also your testes are producing testosterone and DHT, which is dihydrotestosterone. So let's get into what actually makes us male or female, or how do we develop into male or female. So first of all, we have to get into the chromosomes and talk about the genetic material that we're dealing with. So in each cell, we have 46 total chromosomes, 23 pairs of chromosomes, as you can see here on the right, and 22 of those are autosomes that basically code for things like your hair color, your eye color, stuff that's has to do with your physical characteristics. And then you have one pair of sex chromosomes right here, your X and your Y. So notice that the Y chromosome is very small and has less genetic material than the X chromosome. So the presence or the absence of the Y chromosome actually determines biological gender. The Y chromosome produces males. You get one. So if you're getting all of these from your parents, you're getting one chromosome each from your parents. You get one X chromosome from your mom, because she has two X chromosomes, and then the Y chromosome that gets passed down from your dad, because he has one Y chromosome. In females, it would be an X chromosome from your mother and an X chromosome from your father. So, SRY is the sex-determining region of the Y chromosome, which is a gene. So we call it the SRY gene. The SRY gene produces testes-determining factor that binds to DNA and activates three genes: SOX9, WT1, and SF1, otherwise known as Wilms tumor protein and steroidogenic factor. All of these things control the development of the testes. So this gene, SRY gene, has packed in it the genetic material needed to influence other genetic material that will develop the testes. It'll develop that medullary region of the bipotential gonad. It'll develop that into the testes. And so, once we have the male cells that are developing into testes, we have the semiphanerous tuberals, which are right here. And you can see the Leydig cells that are really important, and the Sertoli cells. So these Sertoli cells facilitate spermatogenesis, the production and the development of maturing sperm. And they produce something called, so in the beginning, while the fetus is developing, they produce anti-Mullerian hormone. And that anti-Mullerian hormone actually causes the Mullerian duct that develops into the fallopian tubes in the uterus to digress. So that's really important there. The Leydig cells create testosterone and DHT, which influences more so the development of the Wolffian duct into accessory structures such as the epididymis, the vas deferens, and the seminal vesicles. And DHT that is created by the Leydig cells really influences the male external structures, like the penis, the scrotum, and the glans penis. So, in the absence of the SRY gene, so there's several different situations that you can have where you have an XX chromosome, you can have an XXX chromosome, you can have XO, but typically we have XX for females. And in that absence of the SRY gene, you're not creating the same genes, you're not creating that, it's going to influence those three other genes. So, the testes will not develop. Instead, in the absence, the ovaries will develop. Because the anti-Mullerian hormone that's produced by the Sertoli cells is non-existent. So that Mullerian duct is actually able to develop on its own, given most likely all those genes. There's a lot of different factors within the genes of the two X chromosomes that are received with the female that helps develop the ovaries, the fallopian tubes, and the uterus. So the Mullerian duct turns into the fallopian tube, the ovary, and the upper part of the vagina as well. Also, without testosterone in the mix, Wolffian ducts regress, and without DHT, external genitalia turns into female genitalia, which is the vagina, labia, and clitoris. So that's my presentation, and I hope you enjoyed it. I hope you learned a lot. I'm going to end it there. Yeah.